In addition to this, the endothelium is negatively charged. And because it's negatively charged, it repels platelets. Okay. Um, the endothelium also modulates fibrinolysis. Okay. How? By modulating the profibrinolytics and the antifibrinolytic agents in the blood. Okay. So all in all, the endothelium is a very dynamic tissue. Okay. Let's talk about what happens during an endothelial injury. Okay. So let's try to draw and try to explain what's going on, what happens in an injury. Okay, what goes on in an injury. Okay, so let's say this is uh, this is the representation of an endothelium, and those are the cells. Okay, so let's take one of the cells out and kind of blow it up. Okay, that representing the nucleus, and what is seen is inside there are certain bodies which we call the Weibel palate bodies. Okay. Okay, so these have got a very interesting chemical within them, and that is the von Willebrand factor. Okay? Von Willebrand factor, a very important chemical that is released. Okay, whenever this endothelial cell gets injured. Okay, so this is an endothelial cell. Okay. Okay, and let's say an injury happens. Okay, there is an injury to this cell. Okay, and that injury in turn causes a set of events below. Okay, so we got one cell, and we see there's the third cell out there, and in between we see that there is a damage to the cell, and what has happened is there is an exposure of the underlying collagen. Okay, so the subendothelial collagen is exposed. Okay. At the same time, what happens is there is release of from this injured cell. So what has happened is during injury, so this cell has got injured. Okay, so let's say this is the cell, and it releases the von Willebrand factor. It also releases endothelin. Okay. At the same time, the cell is in proximity with neurons, and so there is also neuronal reflex. Okay. So this events, it results in what? Let's write it down. What happens? What's going on? Okay. So let's write this in sequence. The first thing that happens is vasoconstriction. Okay. So vasoconstriction meaning this blood vessel, it reduces in diameter, it undergoes spasm, and vasoconstriction happens. And what's the reason for this vasoconstriction? There is this neuronal reflex that we alluded to earlier. and this endothelium. Okay. Um, so this endothelium that was released from the injured cell. Okay, let's write it down here. Released from the injured cell. Okay. Then there is this neuronal reflex. So these two things bring about vasoconstriction. That's the first thing that happens. The second event that happens is we see this chemical that has been released from the injured cell, and this cell is this chemical is available in the proximity of the exposed collagen. Okay. So then what happens? Let's draw this here again, and then let's try to show it. So you've got this collagen here, and in the vicinity there is von Willebrand factor. So what happens is this von Willebrand factor, let's say that's the von Willebrand factor, okay, comes and attaches to the exposed collagen. Okay, so von Willebrand factor attaches to the exposed subendothelial collagen. Okay, it attaches to the subendothelial collagen. Okay. So the second event is subendothelial collagen is exposed. Okay, and von Willebrand factor comes and attaches to this collagen. Okay. At this time we need to remember that at this point in time there are lots of platelets passing by, because the blood is moving through this blood vessel. Okay? So the third event that happens is that platelets which are passing by, okay, they bind to this von Willebrand factor attached to collagen, okay, via a receptor that is present on the platelets, which we call GP1B. Okay, receptor on the platelets. Okay, so these platelets which are moving around, they encounter this von Willebrand factor bound to collagen, and they bind via the GP1B receptor. So let's represent that here. So that is a representative of the, so this is the receptor GP1B, okay, and it's present on this platelet. So this is a platelet. Okay, so the platelets bind to this von Willebrand factor via this receptor which is present on the platelets. Okay, then what happens? The platelets, this binding results in the platelets undergo conformational changes. Okay, they undergo conformational changes. Okay. This conformational change, what happens? It results in release of ADP and calcium from the platelets. Okay. This ADP is they help in the platelets adhering to the endothelium. So this ADP makes the platelets sticky. They begin to adhere to the endothelium. Okay. To remember this, we need to remember that ADP and calcium, this is needed for coagulation as well. Okay, so that's a downstream event happening. Okay, okay. Then what happens is very interesting. That's the fourth step. Okay, this ADP. So we're telling this platelet once it attaches to the von Willebrand bound collagen, it undergoes a conformational change and it releases ADP. Okay? This ADP can act on itself and it can act on other platelets and make them sticky to the endothelium. Okay? Okay, so let's imagine that this ADP acts on this platelet. Okay, so there should be a receptor, and that receptor is a P2Y12 receptor. P2Y12 receptor. Okay, so this released ADP acts on the P2Y12 receptor, and that results in a receptor being expressed on the surface of the platelet. Okay, and that receptor, let's represent it like that. Okay, and that receptor is GP2B3A. Okay, so let's recall what's happening. This platelet binds to the von Willebrand factor bound collagen. Okay, and that results in conformational change of the receptor of the platelet, and that conformational change results in the release of ADP and calcium. Okay. This ADP acts on other platelets as well as on the same platelet that caused this change, and that receptor on which it acts is P2Y12. Okay, binding of ADP to P2Y12 results in the expression of another receptor GP2B3A on the surface of the platelets. Okay, okay. So let's write it down here. This ADP, adenosine diphosphate, it binds to P2Y12 receptor. Okay, and this induces expression of GP2B3A receptor on the platelets. Okay, then what happens? Okay. So now we have got this platelet. It has got GP2B3A receptor on it. Okay, and what this GP2B3A does is it binds to a molecule of fibrinogen. Okay, so this receptor binds to fibrinogen, and that fibrinogen in turn binds to another GP2B3A receptor on another platelet. Okay, this again is a GP2B3A receptor on this platelet. Okay, so let's call this platelet 1, let's call this platelet 2. Okay, so the GP2B3A binds to another platelet via this molecule that is fibrinogen. Okay, so let's label it out here. Okay, so the fibrinogen you get a sandwich between one platelet molecule and the next platelet molecule. In between, you got this fibrinogen that's kind of like a piece of cheese or something. An analogy, yeah, so you get kind of like a sandwich. So what's happening is one platelet is getting attached to another platelet. Okay, and there are multiple platelets, and so ultimately what you get is an aggregation of platelets. Okay, right. So the next thing that happens is, um, yeah, so let's write it down here. So the fifth event is fibrinogen binds to GP to B. Sorry for this. Okay, it binds to GP2B, 3A. Then um, that causes irreversible aggregation of platelets. Okay, fibrinogen binds, and this fibrinogen binds to basically two uh, platelets. Okay, right. So yeah. Okay. So now ultimately what we see is that there are lots of platelets which have aggregated, and then there is fibrinogen that's kind of acting as a glue between the platelets. Okay? But this glue happens to be not very stable, and therefore the next part, what happens is the coagulation pathway that converts the fibrinogen to fibrin, and that fibrin threads are relatively stable, and they kind of form a very um, a plug of sorts. Okay. So when injury happens, what we have seen is the mechanism of platelet plug formation. Okay, so let's recollect. Um, the sequence that we talked initially was that the first event that happens is vasospasm, followed by platelet aggregation, and then the platelet plug formation. And those two completes our discussion on primary hemostasis. And we said that there is conversion of fibrinogen to fibrin, and that is the second part of hemostasis, that is coagulation, which we'll be considering in another segment. Okay. Finally, we'll end up with thrombolysis. Okay. So that completes this segment of our discussion.